Thank you for the opportunity to tell you about a company I recently founded called Apira. The mission of this company is to advance small molecule inhibitors of the oligosaccharidyl transferase for the treatment of cancer and other diseases. The oligosaccharidyl transferase is an enzyme that adds N-glycans to proteins as they're elongating in the endoplasmic reticulum. There are two partially redundant complexes that have two different catalytic subunits, STT3A or STT3B. Inhibition causes ER stress, and inhibition also impairs function of highly glycosylated proteins. So why have others not targeted N-glycosylation? The main reason is because there's a natural product called tunicamycin, which is toxic, and so there's therefore perceived toxicity about this approach. However, the targets in this pathway are unclear. Until recently, they have not been fully defined. The biologic effects of inhibiting these targets are not clear, and it's been very difficult to measure inhibition of this pathway. In addition, there are no small molecule inhibitors until what I'm about to show you. We did a high throughput screen of over 350,000 compounds, moved on to chemical synthesis and structure activity relationships to identify this first-in-class inhibitor, which we call NGI1. And you can see that unlike tunicamycin, which is on the end, we get partial inhibition. And this partial inhibition is quite important because it's the reason why we do not see much toxicity with our inhibitor. We went on to validate that there is partial inhibition using knockout models where we've knocked out either STT3 or STT3B, and you can see in the red boxes that we get partial inhibition of both subunits. We also have found that we're inhibiting glycosylation of specific sites, which I don't have time to go into much more to tell you about, but it is a site-specific event. So the initial market we anticipate will be sarcoma, and more specifically osteosarcoma. These are tumors that arise in the long bones and frequently metastasize. The global market for osteosarcoma is approximately $700 million per year. And more importantly, this is a disease with high unmet needs. And what do I mean by that? Well, chemotherapy doesn't work, radiation therapy doesn't work, targeted inhibitors and other agents don't work, and immunotherapy doesn't work. So it's also a rare disease that includes pediatric patients and has a very limited pipeline for drug development. So why sarcoma? We performed a screen of over 500 tumor cell lines to identify tumor types that are sensitive to OST inhibition. Our results shown in this whisker plot indicate that the tumors derived of bone or soft tissue were the most sensitive. And we also found that there are other tumor types such as lung and pancreas cancer that are also sensitive. So we've advanced drug development for this class of inhibitors in collaboration with New England Discovery Partners in Brantford, Connecticut, initially through an STTR grant, through private foundations, and now with Blavatnik funding. These efforts have advanced our potency by greater than 10-fold and our solubility by greater than 25-fold, which has allowed testing in preclinical models. And so the one I'm going to tell you about is NGI-155, which is circled here. So we use this in a xenograft of osteosarcoma, and we found that a relatively low dose of 10 mg per kg causes osteosarcoma xenograft regression. So this is an important finding because osteosarcomas do not typically regress in preclinical models. Our post-treatment pathology analysis of the xenografts show that we indeed are inducing ER stress, as you can see from the brown staining in the bottom here, as measured by induction of BIP, consistent with the mechanism of action. So is there toxicity? We looked at weight, behavior, blood counts, electrolytes, metabolic panels, and cytopathology, and we saw no difference between controls and NGI-155 treated animals. With these proof of principle studies and xenografts, we've now further advanced the inhibitor class, generating about 600 analogs for testing. These efforts have improved our solubility by at least 100-fold and our metabolic stability as well. And so that's our T1/2 is about 50 minutes in microsomes now. So our future goals are to identify leads with optimized in vitro ADME properties, perform in vivo PK studies, and then do our complementary in vivo tox and efficacy studies with the goal of filing an IND application. Thank you very much for your time.